This is CheloniaCast, episode 39. Today we are joined by Dr. Juliana Sterli, who is a researcher with the National Scientific and Technical Research Council of Argentina, based out of the Museum of Egidio, Feruglio. Dr. Sterli has done a lot of work with fossil turtles and turtle paleontology, as well as global turtle phylogenetics, and more specifically with the biogeography and phylogenetics of the Pleurodires and Chelid species. So we're really excited to talk to her today. Thanks for coming on.
Oh, thank you very much for your invitation. And it's an honor to participate. Thank you.
So, Dr. Sterli, what first got you interested in turtles, and what made you decide to pursue studying turtle biogeology and evolution?
Well, since a little child, I was interested in paleontology. So I decided to study paleontology at the university. And when I was studying, I was interested in doing field work. So, and my first field work was in 2001, so more than 20 years ago. And in that field work, we went to a Middle Jurassic outcrop in Patagonia that is very famous. It's Queso Rallado locality, where you can find many small vertebrates. And among the fossils that you can find in that locality, turtles are very, very abundant. So you can find different parts of the shell, and even skulls that are very rare in the fossil record. So I was there, and the leader of the fieldwork was Guillermo Rocher. He studies mammals. So he offered me to study those turtles that were really, really nice with skulls and almost complete shells. So I started to get involved with turtles, and when I came back to the university, I contacted Dr. Marcelo de la Fuente, and he is a leader in the study of fossil turtles in Argentina and South America, and he's also known globally. So I started contact with him and studying these turtles from the Middle Jurassic, Queso Rallado locality in Patagonia. And after that, as I was in my last years of the university, I decided to study those turtles more in detail, and I did my PhD on those remains that now they are known as Condorchelis antiqua.
That's real interesting, and it's looking at some of the amount of fossil turtles that have come out of sort of southern South America. A lot of it from your work and others that work close with you it's really interesting, and it's helped a lot in terms of contextualizing how turtles are related in general. Maybe we can sort of start with, because you've also done work with the global phylogeny of turtles, and in order to sort of segue into that conversation, it's good to just talk about how a phylogeny is constructed. And with turtles in particular, it's been sort of challenging. There's unique challenges, and you can speak to that. But I'm curious, sort of, what do you think about using molecular versus morphological data to construct turtle phylogenies? What are the advantages of both different sources of data, and sort of how is that relevant to the question of turtle relationships?
Well, I think that it's very important to consider all sources of evidence to look for the phylogenetic relationship of turtles. Of course, maybe the most common for us, for paleontologists, is morphology, because it's what we get in the fossil record, mainly the bones. But as turtles have extant species as well, they live today. I think it's really, really important to consider also the information coming from molecules and also from the soft part of the body, as well as other characters that could be used as ecology, ethology, like many other sources of characters. As I'm a paleontologist, of course, I'm more used to morphological characters from hard parts, as the shell and the skull bones and so on. But I also consider very important to include these molecular data that are usually they are available in GenBank or other sources. So I would use both data when, for example, I'm working with some group of turtles that have extant representatives. For example, chelids. I try to use both set of data. Sometimes it's really difficult to use both, I would say for Testudines, for all the crown group, is really difficult because the molecular data have a certain signal for the relationships of turtles, while the morphology has another signal. So when we put all together, they could create some, between brackets, weird results that are not expected. So yeah, if you can, I would suggest to use all the data that is available, but sometimes is not possible.
Right. That's interesting to think about, and it's just getting more, and that's where sort of a lot of molecular analyses or morphological analyses make a lot of assumptions about how turtles are related and just use extant species. But you have come in and used fossil species, and then you have to use a lot of morphology for that, and that can it's a good thing, it seems like, for phylogenetic analyses to include fossils, but it also creates more challenges. You mentioned some issues that can arise, and I'm curious what's sort of the major issues based on different types of data in the turtle phylogeny are that you've come across.
Yeah, well, for example, two clear examples of these different results that we can get using morphology only or molecules only in phylogenetic analysis is the position of, for example, the long-necked chelids. If we consider only molecular data, we will see that the chelids from South America form a monophyletic group, while the chelids from Australasia form another monophyletic group. And each of these two clades have a species with long necks. While if we consider the morphology, in morphological phylogenetic analysis, we will get that these long-necked chelids form a monophyletic group. So this monophyletic group has species from South America and from Australasia. So you can see two different hypotheses and two different evolutionary histories in that. And the other very usual different hypothesis in turtle evolution is the position of Trionychians among cryptodeers. If we consider the molecular data, Trionychians are the most basal clade of cryptodeers, while if we use only morphological data, Trionychids are well nested inside the cryptodeer clade. So two sources of evidence, two different stories about the evolution. That's why it's good to put all together to have only one. But sometimes it's difficult.
Right. And the current consensus with the Trionychia, what do you think the most likely relationships are for that group? That's sort of a dilemma in turtle evolution.
Yeah, well, if we see also the fossil record, there are some authors that consider some turtles from the middle late Jurassic from China belonging to this lineage of turtles, the Trionychians, and they are among the oldest cryptodeers. So if you consider that also a source of evidence, paleontology would say, okay, maybe the oldest cryptodeers are belonging to this lineage. But again, I think we should make a total evidence analysis to see really where they belong. We have tried to do that, like a total evidence analysis of testudines, but it's really, really difficult. The relationships that we get are very unorthodox, so we are keep on working on that to see if we can improve the result and include more sources of evidence.
Yeah, that sort of raises a lot of questions in terms of just where different clades arose along the tree. Something you pointed out in one of your papers that I found pretty interesting was that and you spoke a bit to this by saying that actually we haven't really touched on this, but the pleurodires, they could be nested. You could have a paraphyletic cryptodyra, and that seems like it sort of goes against a lot of just assumptions of previous studies. And I'm sort of curious, what are the odds of that being the case, and what sort of lines of evidence are there for a paraphyletic cryptodyra versus which is generally assumed monophyletic cryptodyra?
Well, yeah, that's one of the problems of that. The things I'm saying about analyzing molecules and morphology all together in a phylogenetic context. Unfortunately, maybe the morphological phylogenies, this more like a global phylogenies, do not have yet as much characters as we need to solve this issue. Although maybe with molecules we have more data because the sequences have many characters, although not all are informative. Okay, we don't have so many data with morphological data yet. So when I did that paper about this total evidence analysis in turtles, I pointed out that in that case, when we put all the information together, pleurodires were nested inside cryptodires, but I think that's more an artifact of the analysis and the methodology I used, that is parsimony, rather than a real result or history. We need to consider that each method has its own flaws, and
right.
We need to consider that, and we need to see, okay, nowadays we have other methodologies as Bayesian analysis or maximum likelihood, maybe to analyze molecules, and also to include morphology as well in those models. So nowadays, maybe we have other methodologies to analyze that data, because that was like a while ago, that paper. So I think it's more that, and if we consider the relationships in an unrooted tree, as I showed in that paper, we get more or less the same interrelationships among the extant clades, among I would say the largest extant clades. And it seems that's related with the root that the crown clade gets when fossils are included. So it seems the inclusion of fossils in this total evidence analysis modifies how this clade Testudines is rooted. So in that, it changes the root of the tree, and pleurodeers get inside cryptodeers. But okay, nowadays maybe we can analyze it better with more morphological data and with newer molecular data and another methodology maybe, and get different results. So
right, that it sort of gives a different, including the fossils. It's really interesting to look at because you see the split between the two major suborders, or Pleurodira and Cryptodira, is a lot further down the tree than you would assume. And you've got a lot of fossil groups of turtles that are outside of that sort of diversification. Sort of curious, there's a lot of different species. You've worked with melanoforms and other groups of turtles, and those sort of all seem to fall outside of the split between Pleurodira and Cryptodira. Where do those groups fall, and what are some of the major fossil turtles in that assemblage of stem turtles?
Well, yeah, the thing is that the knowledge about turtles and their evolution have changed maybe during the last, I would say, 40 years. Maybe 40 years ago, we had like an idea that there were two clades, and all living and extant turtles were belonging either to Cryptodira or to Pleurodires. But during the last, I would say, 20 or more years, new fossils were found, and also new interpretations of old fossils appeared. And with the cladistic studies and the use of computers to do cladistic studies, this like a previous idea about these two groups and has changed a little bit. So now we know that we have these two main clades, Pleurodires and Cryptodires, but we also have many, many other groups in the stem that are outside this, what we call the crown group, Testudines. So among them, we have these Triassic turtles that are the oldest that we know, Progynochelys, Paleocercis, Proterocercis. And then we have other turtles along this stem. We have Cayentachelys, that is from the US. You might know about it. But before, it was considered a cryptodire. Now we consider it belonging to the stem. And among these turtles from the stem, I'm focused in the evolution of Meiolaniforms that are, I would say that the most famous Meiolaniforms are the Meiolaniids, that are these terrestrial giant turtles that have horns, and also the tails are covered by tail rings, and they have also a tail club at the end of the tail. So, and again, these Meiolaniids before were considered cryptodeers, but now we know, or we think, that they belong in this stem. So this stem is really, really diverse.
It's interesting that it's so diverse, but that a lot of those families or groups, I mean, they're pretty specious, and they're no longer around. For it's sort of a context dependent question, like family or clade specific. But what are some hypotheses you have about how they went extinct?
Well, there are like different hypotheses for different groups, because these groups or these turtles went extinct like in different times. Yeah, because these from the Triassic went extinct in the Triassic, and these Meiolaniids lived until like kind of recent times, right? Like to the Pleistocene, even the Holocene. So there are different hypotheses for each group. In general, we can say that these, of course, climatic changes played a role in the extinction of many clades, right? But we should know which particular clade we are speaking about to see if we have another like kind of explanation for that. But I would say that climatic changes could be a source of these extinctions for many clades.
Interesting. Yeah, I know Jack is pretty interested in that the Meilaniids, and I'm curious, like, what do you think about the odds of people for that group specifically playing into the extinction of those? Because they were, I mean, they were in areas where you had settlements and humans.
Well, yeah, there was a paper saying that Meiolaniids were living with humans in this area of Vanuatu in the southwest of the Pacific. But actually, we have seen the publication, and we consider that those fossils they are not Meiolaniids. They are actually testudinids. So they are tortoises, basically, that they live today. So that's the only settlement where they can kind of prove that these turtles were together with humans. But in our opinion, they are not Meiolaniids. They are tortoises, testudinids. So until now, there is no evidence of of humans and Meiolaniids being together in the same time. So it seems that Meiolaniids went extinct before the arrival of humans in Australasia.
On the topic of the Meiolaniids, there was no evidence that they coexisted with humans on New Caledonia at all either. Like are those the same fossils you're referring to? Like those were tortoise fossils throughout most of those islands?
Yeah, no. There is no evidence so far. I can share a paper that we did. It's more like not a scientific paper. It's more an outreach paper that you can. And we say we discuss all these, and it's interesting because we have this idea that they lived together, but no, some of the fossils we cannot identify if they belong to tortoises or Meiolaniids in some cases, but in those cases that we can identify the fossils that are for example, femora that are very characteristic of testudinids. So there is no doubt that they are testudinids. So, yeah, I think the idea of humans and Meiolaniids, it's much cooler with testudinids, but unfortunately, they didn't meet, at least what we know so far.
I think that's going to be one of the best pieces of information to come out of this podcast, at least for some of our viewers are really going to appreciate that. But kind of taking it in a different direction, still on the topic of Meiolaniids, I was reading one of your papers. I found the hypertrophied nasal cavity to be really interesting, and that it may have had implications for it was the climate, not necessarily like olfactory unit a bit.
Yeah, well, we have scanned these skulls of Meiolaniids, a species from Argentina and one species from Australia. And we have seen that when we reconstruct the inner cavities of the skull, we can see that the nasal cavity, it's really, really expanded, really big, compared with the rest of the I would say, the brain and other cavities. So it was like very we were very surprised about that and said, okay, let's compare with some ecologically equivalents that could be the testudinids today. So we also scanned some testudinids, and we also saw that the testudinids also have like a big nasal cavities in their, I would say, in their heads, and they are really big compared with other turtles that are not terrestrials, right? Marine or aquatic turtles. So we were trying to see if there was a correlation with the environment or with the behavior, maybe, because the nasal cavity has receptors for smell, of course, and also it's related with breathing. So it has many functions in the cavity. Yeah. So we wanted to see if there could be a relationship with environment or with behavior. And we said that in other reptiles, when they have these big cavities, they are related with that they live in more arid environments than other species from the same group living in more humid environments. So we were saying that we don't know, we don't really know. We can hypothesize that they could be related with a more arid environment, because by that time in Patagonia, the climate was going to be a little more arid than before. There was a climatic change in Patagonia by that time, and also in Australasia. So we said, okay, could be that, and it could be also related with some behavior, like, for example, this mating behavior that the testudinids have, that they smell at each other. They also have some, they can be like hit each other. So we were also exploring that possibility, that, okay, maybe it's related with the sense of smell of these turtles. But okay, as we don't have the soft parts, it's difficult for us to test those hypotheses. So we are like in this phase that we are exploring and comparing and suggesting some possible hypotheses about this enlarged nasal cavity.
It's a little. I just got back from Belize, where I was doing some field work, and the sliders there, the males have these really pronounced bosses on that's more of the external nares. But you're saying that like the internal nares is expanded. So what? Yeah. I mean, the sliders, it seems like it's sort of a mating thing because it's sexually dimorphic, but this could be something else if it's more internal. Yeah, that's really interesting. You've done a lot of sort of cutting edge or been a part of projects doing pretty cutting edge analysis of fossil material. One thing that was pretty interesting going through your work is you've done some like neuroanatomy, where you can scan a skull and construct you can get a reconstruction of how the nerves were wired. I'm curious how that's informed our understanding of fossil turtles or fossil chelids.
Well, it's a good question. The thing is that if we compare the amount of data that now we have about the neuroanatomy of turtles, I would say more like based on fossil turtles, is still little if we compare the information that, for example, we have from dinosaurs. So I think we are still in this phase that we are gathering information in different groups. And I guess maybe in the next years, we will have a better idea about how informative this, the neuroanatomy of fossil turtles is. With the information that we have now, we can see that, for example, they are pretty conservative in general. We cannot differentiate yet between different morphologies, for example, in the inner ear between marine or terrestrial turtles. Although you can see some differences, they are difficult to measure yet. And we cannot say 100% sure if an extinct species was marine or terrestrial based on the inner ear. So there we need more data. We still need more data to figure it out. On the other hand, it seems that we can see differences that can be transformed in phylogenetic characters, and to see if they are useful in phylogenetic analysis. We haven't done yet that. So I think that would be the next step, to gather more information about more extinct and also extant turtles. Because what the problem with this reconstructing the inner cavities of the skull in reptiles is that the brain and other soft tissues in the inner part of the skull are not like 100%, or they don't reflect 100% the shape of the inner cavity. For example, the brain has not the exact shape of the cavity. So what we are reconstructing is not exactly a brain, because there were many other tissues around. So we need to scan like extant turtles that they have the brain and other tissues associated, to see the relationship and the correlation of the brain and other soft tissues in this inner cavity. So there are many questions to be answered before exploring, for example, the ecology or phylogeny of these inner structures.
Can you get, I believe it's either 12 or 14 primary sensory nerves that the turtles have in the skull. Mm-hmm. So that's conserved through fossil turtles, or are there any universal features you see in fossil turtles with regard to the nerves that are just different than modern extant turtles?
No, I would say that the general bowel plan is the same. What is different, but unfortunately, there are not so many good fossils to test that, is that, for example, in one specimen of Proganochelys, this Triassic turtle, that is one of the oldest, and in Cayentachelys from the US, it seems they had another bone that is not ossified anymore in extant turtles and many other fossil turtles, that is the laterosphenoid. That a bone that is located in the, I would say, like in between the orbits, more or less, in that location in turtles. And that bone was, through that bone were going out some nerves that we cannot see in any other turtle. So it seems that these more basal turtles, they had this laterosphenoid ossified. So that was different. But unfortunately, we know only those two cases, and they are not so well preserved to be explored in more detail. But I would say that would be the main change that occurred. That in I would say, turtles from the Middle Jurassic onwards, they do not ossify this laterosphenoid. So the nerves that are going out and in that region of the skull, we cannot trace them in these neuroanatomy reconstructions that we do in the fossil turtles.
That's really interesting that you can go back and get that detailed. I mean, you have some level of uncertainty, but that you can essentially use the holes in the skull where those nerves and veins and such would pass, and you figure out the structure of those things. Really fascinating. It's interesting to think about it. So you mentioned you've also done some work with the basis sphenoid, the parabasis sphenoid complex, and that we had a previous guest on that talked a bit about the evolution of the turtle shell, and how there's different types of ossification that have formed a union, and that's allowed for this interesting evolution that's not seen in other vertebrates. But there are some kind of parallels to that kind of ossification in your work with the parabasis sphenoid complex. Maybe you can talk to that and why that structure is interesting.
Yeah, well, the parabasis sphenoid is interesting because we use a lot in the cladistic analysis about turtle evolution. How is the location of the entrance of the carotid arteries in the skull of turtles? So it's a character that has been used many times, and it was like having a lot of attention. If the carotid entered in the basis sphenoid or in the prootic or in the pterygoid, and the main clades, I would say like cryptodeers and pleurodeers, were defined by that, and also the paracryptodeers and the basal turtles. So as I was working on this phylogeny to see if Condorchelis was a cryptodeer, as Gaffney was saying about Cayentachelis, because they were very similar, or if it was more basal. I was exploring a lot about the carotid characters. So looking to fossils and to extant turtles, I was like seeing that when I was looking at the basis sphenoid, it appeared that it was actually two bones fused. And in some fossil turtles, it was really, really evident that it was like a more like a squarish bone, a thick bone, that it was the basis sphenoid, and below that, it was more like a elongated bone, thinner, and it was kind of different. So I started to try to see that in more detail, and we micro CT scanned skulls, and we were looking that in more detail. Well, and we came with this hypothesis that it was actually that the basisphenoid was actually formed by the basisphenoid and also the parasphenoid, forming this parabasisphenoid. And as you were saying, the basisphenoid is a autostossic bone that is formed. Before it was a cartilage and now it's a bone, but the parabasisphenoid is a membranous bone. So it's a mixture of two kinds of bone in one. And we also hypothesized that the development of this basisphenoid trapped the carotid artery, mainly in the, I would say, in the cryptodira lineage, and moved the entrance of these carotid arteries towards the posterior, towards the pterygoid and towards the posterior part of the skull, like giving some protection between brackets to this very important artery that gives all takes all the blood to the skull, right? So we proposed that, and we have also explored how was the case in other vertebrates as well about this artery, because it's a really important character.
It's really interesting to analyze specific morphological characters. It's one thing to create a matrix of 150 different morphological situations and then just use that in an analysis, but it's another thing to actually look into all of those characters and how those things have changed and what the functional significance of those characters is. And in this case, how the character changes has changed other aspects in terms of the arterial anatomy. It's really fascinating. That sort of brings it back to a question that I ask a lot in terms of like how people when you're constructing like a morphological phylogeny, you assume that there's certain assumptions that underlie the test to throw out certain characters that are homoplacious, and then it narrows into certain things that are synapomorphic that are informative for different groups of turtles. I'm curious how does that process work in that analysis? How do you know what characters are informative versus which are not?
Well, you don't know a priori if a character is informative or not, because that you will test it when you run the cladistic analysis. Okay, so the first step is to build your matrix to find the morphological characters, and that's a very like comparative anatomy. Plain comparative anatomy, like you see, okay, this neural one in this case contact costal one only, and okay, but there are some other turtles where neural one also contacts costal 2. So okay, I will propose a character that is neural one contacting costal 1 and neural 1 contacting costal 1 and 2. So first you create the characters, and then you see, of course, the characters are related with your sample. If you are only analyzing one family, you will try to see how diverse your family is and which characters can be used for that family. So you narrow your study, you define your characters, and then you run the analysis, the cladistic analysis. And that's where the result will tell you if some characters are synapomorphic or not for a certain group. So a priori, you don't know. It's just, it's what we say, like is the first step to establish homology, right? It's comparative anatomy. But then the second step to see if those characters are homologous or not is the cladistic analysis. If you run the analysis and you see that, okay, this character, neural 1, contacting costal 1 and 2, is only present in one clade, you can say that it's a synapomorphy for that clade. But beforehand, you cannot be sure. And a character that could be synapomorphic in one context with a more, I would say, reduced sample cannot, maybe when you include more species, is no longer synapomorphic for that clade only, but could be also a synapomorphy for another clade. So it depends the context as well.
Yeah, that makes sense. There's sort of variable, different techniques that have been used through time to analyze how phylogenetically informative characters are. It's sort of, that's an area of development, but that it's something that's more it's borne out through analysis and not personal decision. That's something that's more of a sort of a analytical artifact. Yeah, so okay, that makes sense. I'm sort of switching focus a bit more, and maybe going back to the major turtle phylogeny. You've also done some work with dating different splits, specifically the split between cryptodires, pleurodires, the chrontestodines. And you found there's some conflicting results there with molecular and morphological and total evidence phylogenies. I'm curious why you think that there are what, first of all, what have you found in that area, and why do you think there's incongruence between the different lines of evidence?
Well, sometimes it's the evidence that each researcher uses for dating a tree. For example, sometimes if you are a molecular biologist and you only consider the molecules as evidence, and you go and see and search in the fossil record, which is the oldest, I don't know, Trionychian. But you consider, okay, this is the oldest Trionychian. I will put in my matrix, this is the oldest date for this clade. But maybe no one has included that species in a cladistic analysis. So you don't know, actually, if that species is a Trionychian or not. So maybe they use a fossil that is no longer considered a Trionychian to date that node, so they can have a different result. So that could be one of the problems. They could use, it's usually related with that, like to use fossils that are not included in phylogenies. So you don't know exactly which node this fossil is dating. And then you have, of course, differences based on different methodologies. If you use molecular clocks, you will get some results different if you use, I don't know, this, I would say, by hand, to calibrate the tree by hand based on the taxa you have in your phylogeny. So there are differences like in the taxa you include, in the phylogenies, and also sometimes the ages of the formations where the fossils are found are not accurate. I don't know. In the original paper, it was published as this fossil coming from the late Cretaceous, but I don't know, 5 years later, they do like a dating of that formation, and they say, oh no, actually is late Jurassic. So you have a difference in time. So I think there are many different things that could lead to different results. So when we compare different results, we need to take all that into account.
Right. Yeah. And so in terms of the actual times for the split, that's one that's there's a lot of variability on it. And it seems like you found that maybe it was a bit later than expected. Maybe you can just shed some light on that.
Yes. Well, if we, in general, I would say, like really general, when we use molecules to date, to calibrate trees, we will get older dates than using the fossils or morphology because of the methodology and the algorithms they use. In this methodology that we developed, our main concern was this about the phylogenetic position of the species in the trees. So, for example, as I was telling you before, sometimes with the fossils, what we have is that the fossils are fragmentary. Maybe we don't have the whole animal, so we cannot score the whole animal in our matrix. So that taxon might jump from one position to another. So maybe in one analysis, one turtle is inside Trionychia, and sometimes it goes outside. So if you use that fossil to calibrate Trionychia, well, maybe it's not so good because in some analysis it goes outside. So we wanted to test that in that research. So we were exploring this bootstrap uncertainty range to see if we pick a certain node and explore the ages that were calculated for that node, considering or not some species inside. So we didn't give like a only one date for a node, but a range, a minimum and maximum range of ages for that node, because sometimes you have some fossils that go in and out of a specific clade. And with that, we consider that it would be useful to use it when fossils are included in phylogenies, and we want to calibrate and to see when a node was originated. So with that, using bootstrap replicates, we can explore the age of the node. So that was different from other methodologies, for example, molecular clocks, or even this that to calibrate by hand, I would say.
Incorporate some other areas of uncertainty in the analysis. Yeah. It may be a good time too to just in phylogenetics, there's kind of a statistical a test of how variable the statistical the most likely tree is, and that's the bootstrap or jackknife values. Maybe you could, for someone that's not familiar with it, that may be a kind of a foreign concept. Maybe you can explain what that is and why it's so low usually in turtle phylogenies.
Yeah, well, these bootstrap and jackknives are resample methodologies to see how well supported a clade is. So we run our cladistic analysis using parsimony, or you can use TNT, or the other, what is the name? I don't remember now. I use TNT usually. So you run reanalysis and you get your most parsimonious trees. But you don't know exactly how well supported your clades are. Okay, they are supported because, of course, you have some characters supporting those clades, but you don't know if one clade has more support than other clades. So after running your cladistic analysis, you run this bootstrap or jackknife resampling that is not a cladistic analysis. We need to keep in mind that it's not that we can use those trees as evolutionary trees. Because I've seen those kind of trees as examples of evolutionary trees, but they are not. They are just, they give us some information about the how much information we have for certain groups. So once you run the analysis, the bootstrap, you will get a resampling of the characters, like, of course, randomly, and you will create a new matrix with the same amount of characters that your original matrix. With Jackknife, you will get a smaller matrix because it's not with I would say, with the replacement of the characters. So you will get new matrices, and then you will make this. You will run again the analysis, and you will get numbers, as you were saying, and you will put those numbers as measurements of support of your clades. And of course, they are from 0 to 100, and you will put all the numbers in your nodes. Why we get so low bootstrap or jackknife or even Bremer supports in our trees is mainly because we are incorporating fossils. As I was telling before, fossils usually are not found complete. So we have many missing data in our matrices. And
Right.
in the resampling, when you run the resampling, sometimes you don't get the clades because the characters that were sampled were not including the taxa that we wanted. So that's why we get so bad bootstrap, jackknife, and Bremer supports. If we run the analysis only with extant taxa, for sure those values will be much better.
Yeah, still not perfect, but higher because like you said, you got certain fossils where you've got really fragmentary material, and that sort of makes the confidence go down on certain relationships. I think maybe we can shift focus to it's kind of interesting looking through your work. You've done work with sort of the global turtle phylogeny, and then specifically with Pleurodira, and then with Chelidae, Panchelidae, specifically within that clade. So it sort of goes down the tree, which is cool. But maybe we can shift a bit of focus to Pleurodira and sort of biogeography of that group. Sort of a fascinating group of turtles, kind of the lesser species group of all turtles, but a rich fossil history and biogeographic history. One that's real interesting with that is the Rhymnochelys, Peltocephalus, Podocnemus group, and just how they got into the situation they're in biogeographically. What do you think? What is known about that group and how they're related, and what events led to their current distribution?
Well, that's another example of maybe differences between molecules and morphology that we were talking about before. Well, lately it's the latest, I would say, molecular analysis about the Podocnemidids show a close relationship between Podocnemis and Erimnochelis, as forming a clade, and outside Peltocephalus. So if we see that, maybe it doesn't, between brackets again, doesn't make sense from a biogeographic point of view. But okay, molecules are suggesting those kind of relationships. So, of course, what we need to do first is to run the phylogenetic analysis and then evaluate the biogeographic history of a group, okay? Because biogeography itself is not a character in an evolutionary tree. It's just the location of the species. So this is secondary evidence. So we need to focus on the phylogeny. And in this case, it's weird that, okay, the South American podocnemidids are or have inside this species from Madagascar. So we need to explore what happened there. Maybe it was a dispersal because South America and Africa and Madagascar were separated well before. It was around 110 million years. So it's really, really far away, the split of those continents. And this molecular dating, they are saying that the split was in the late Cretaceous, that is much later than 110 million years ago. So I think we need to consider, like, first the phylogenetic evidence, and then try to see the biogeographic evidence. And of course, in this molecular data, they don't consider fossils. So it will be good to make a total evidence analysis to see, to include fossils and more morphological evidence, of course. So yeah.
So it's kind of an open question. The relationship there.
Yeah. I would say that most are open questions. It's that's, I think, fascinating about I would say research or science in general, that we don't have the last word in any of the things. They are just phylogenetic analysis. They are just hypotheses. So if we include more evidence, there would be, I don't know, new fossils or new characters. We sequence new molecules and we include it again in a bigger analysis. We can get new results, and that's fine. I think we should keep that in mind, that a phylogenetic tree is just a hypothesis. So it should be tested with a new analysis.
Right. So with the Pleurodires too, the morphology, I know there was, I forget who the author was, but at some point, and I don't, it's not the case now, but at some point there was some debate about whether Pleurodire was paraphyletic or not monophyletic. Some of the characters that were are used with that group a lot, like the length of the vertebrae and such, and the movement of the joint, the jaw adductor, and the skull, that kind of thing. Those were not informative characters, but that's sort of outdated at this point.
Well, it's in this that I was saying before about the chelids and the long-necked chelids, there is still a debate, because if these long-necked are synapomorphic for a clade, or if they were developed in parallel by two different clades, like this Chelodyna in Australasia and Hydromedusa and Chilus in South America. So I think they are still like open questions related with this, with the chelids. And with pleurodeers, there was also like some of these basal turtles, Paleocerces or Proterocerces, that were considered in the lineage of pleurodeers because they were described as having a sutured pelvis to the shell. But nowadays, with this new analysis, we see that, for example, Proterocerces has a sutured pelvis, but they do not belong to the lineage of pleurodeers. They are much more basal. And Paleocerces, we studied the specimens, and now I can confirm that the pelvis is not sutured to the shell, so they are basal as well.
Okay, so it's almost like it probably wouldn't be right to say, so that is an informative. The pelvic fusion is something it's an informative character for the monophily of pleurodires. But if you take it with respect to stem turtles, it becomes homoplasius. It wouldn't be useful.
Well, it's useful anyway, because you will have it as a synapomorphy for pleurodeers, and you will have it as another character. In this case, a synapomorphy for Proterocerces as well.
Okay.
So it could be a synapomorphy like in two different places in the tree, but okay, those characters are homoplastic. Okay, they evolved independently in those two clades, and maybe you need to see more detail if I don't know if they are exactly the same. How the pelvis is sutured, or how the pelvis or the shape of the pelvis is, if it is the same or not. So they might not be exactly the same, but if we see the character sutured pelvis, it will be like a scored present for pleurodeers and for Proterocerces.
That brings up an interesting question too. How much does nuance go into the character for morphology for fossils? Like you said, it could be there could be slight differences. Are there a lot of characters now that you think are scored as the same thing but are actually slightly different?
Yeah, well, the thing with the matrix or any cladistic analysis is that you revisit the matrix all the time, because I don't know, a new fossil is discovered, or you have seen another collection that you haven't visited before, and there are fossils that you can observe firsthand, or extant turtles that you can observe firsthand, and they can change your interpretation of some characters. So I would say that it's a good practice to come back to the matrix and to critically see the matrices and the characters, and to see, okay, this character is scored as the same in pleurodeers and Proterocerces, but is actually the same or not? Are there differences? Can we see maybe to split this character, or is it useful this way? So I think it's something that we do all the time. When we work with a new turtle, we usually revise the matrix and revise the characters. And sometimes you have a new species that, for example, you have a character well defined with two states, and your turtle has neither of those two states. So you need to add a new state. So yeah, I think it's healthy to do that and to revise all the time the matrices.
That's I think good advice. In kind of going into one more sort of topic, and then we can kind of wrap things up. But talking about, I'm curious first of all what you think if what the most likely scenario for the long neck and short neck is it they're mostly related on each continent, or is it long necks together and short necks together? What do you think is the most likely?
What a hard question. The thing is, when they you are not the only one that asked those questions to me, but also with the origin of turtles. And for me, I'm not stick to one of the two hypotheses. It's not like, oh, I believe this is the answer, and I will pursue. I will look for the characters to be like that, and no, otherwise it's not correct. No, I think it should be the other way around. We should to see the turtles, to see the morphology, to see the molecules, to see the fossil record, to see the extant ones, and try to put all the information together and incorporate it in a matrix and run the analysis. Because if we have a preconceived idea about how the evolution was, for sure you will look for that to be true. And I don't like that kind of, like I would say, way of thinking. I just prefer to try to be, because it's difficult as well, try to be as, not to have an idea in my mind, just to see the morphology and get as much information as I can and test it in, for example, in a phylogenetic context. That's how I think, and I try to be like that, not to have a preconcept of evolution in my mind and to fit the evidence in that preconcept. I don't know if I explained myself, but yeah, I try to do that.
It's like, so it's sort of, you don't think that there's enough data necessarily to say one thing or the other, and yeah, that's sort of, you don't want to introduce bias, right?
Yeah, well, nowadays I'm working in a total evidence analysis of these chelids, gathering information, and for me, we don't have yet a large morphological matrix for chelids. They do not include many characters, and they do not include enough extinct and extant taxa. So every time that we add a new species, mainly extinct species, the phylogeny breaks, because it's a disaster. So I think we don't have, the phylogeny is not stable yet. So I think we need to look for more information. So I'm doing that nowadays to gather more morphological information on extant chelids, and also including extinct, to see if we can make it more stable. And if we can make it more stable, to include also molecular data and have like a total evidence analysis. Because nowadays it's really, really unstable. Both. No, both. Sorry, the molecular is very stable with these two clades from the two continents, but the morphological is still really, really weak. Yeah.
Yeah, and you want those lines of evidence to converge. So that's future project or current
Yeah.
ongoing. All right, exciting stuff to look forward to. Sort of the last thing, like you said, you do the phylogenetics, but then you have to sort of contextualize that with the biogeography. And Gondwana and the breakup of Gondwana is the thing that's hypothesized to have driven a lot of chelid specific evolution. But then when I'm thinking about that, it's like, okay, that's interesting, but it's so broad that it's almost not interesting. But in a book that you co-authored on the evolution of clerodires and South American turtles, you go in pretty good detail with the uplift of different mountain ranges and volcanic activity that could have led to the diversification of the chelids. How much is known about those specific things, and are there any specific instances in the history of Gondwana that maybe had a disproportionate effect on the diversification of chelids?
Well, the main hypothesis is that these Phelomedusoids were originated and diversified in the north part of Gondwana, while chelids were diversified and originated in the southern part of Gondwana. And there are some studies that define that between these two regions there were a big desert and also some basaltic rocks that are more or less coincident with this origin of these two clades. So that could be one. And then, of course, we need to see how is the history about these chelids, if they are actually two monophyletic groups or representing the two continents or not, because that would involve different processes and different ways of distribution, right? So if we don't have a stable phylogeny or phylogeny that, I would say stable, that stable means that you run new analysis and the topology remains more or less the same when you include new species or new data. So with the chelids, maybe we are not there yet. So it will depend on the topology of the tree. But of course, in this southern Gondwana, we have the separation of South America from Antarctica, that it was in the Eocene, more or less, between the Eocene and Oligocene. But Antarctica from Australia was separated well before that. So the history, when all these continents were together, was more or less in the Cretaceous. So it's a long history that Australia is not connected to Antarctica. So if we consider that the South American chelids and the Australasian chelids are two groups, the evolutionary history of this is separated since many, many million years ago, like from the late Cretaceous.
It's really interesting to think about how if there was just the evolution of long and short neck turtles is fascinating to me. It's like for those two conditions to arise independently, if that was the case, it seems like it couldn't be completely mutually exclusive. Like there has to be some kind of inertia in those groups. They split, but there was already something going on where you had a divergence, and then it sort of duplicated when you had them split between the different continents once they were separated. But that's upcoming analysis. I think that's sort of I think we've covered a lot of things, and it was a really interesting discussion. We liked at the end to do a little turtle trivia, like volley. I forgot to mention this in my email. I've been doing that lately, but it's just a way to bring in obscure turtle facts. If you have some random turtle facts, fossil turtle facts, or anything, and you want to toss some our way, we can try to answer them, or we can toss you some questions, or we don't have to do it, but just kind of a fun way.
I don't know.
Yeah. Normally, I like to tell people in advance. It's tough to put them on the spot, but
yeah, because now I need to think about. If you want, we can do it later, and I can send you some. And I don't know if there is a space to do it, that people can do it, or you can do it.
Yeah, I mean, yeah, we can.
Format.
Yeah, we can do that. If you send me a list, it's sort of I don't think we really have a format for this, so I could literally just at the end attach the questions. People can read them on the screen and then try to figure them out for themselves. If they can't figure it out, search it. Yeah.
Yeah, we can do that. You tell me, and have no problem.
All right, cool. Yeah, we'll keep in touch about that. I'll get that. All right, that sounds good. Well, Jack, unless you have anything else you want to go into, or
no, I'm good today. This is really interesting. Thank you for sharing with us.
Yeah, thank you, Dr. Sterli. It's been awesome. I'm same boat here. I've learned quite a bit. It's real interesting to think about a lot of stuff, and I admire your sort of objective position on a lot of things. Like, you're not quick to jump to any conclusion about something, which I think is really good for scientific reasoning. So yeah, thank you for coming on.
Thank you for the invitation. It was really nice to talk to you about turtles. And of course, fossil turtles are like my focus and research, but I enjoy speaking about turtles in general. So it's been great. Thank you.
Cool. And we have one more question we like to close with. If you have one piece of advice for someone looking to make turtle research part of a career, what would that be?
Well, I think turtles are really, really interesting topic to do research, and there are many, many things to be done. Of course, I know more about, and I will focus more about fossil turtles and phylogeny. So I think there are many interesting topics to develop. If you like more like anatomy, you can do to explore different groups. There are many groups of turtles that are understudied. So I think if someone is passionate about turtles, or they want to do research in general, I would recommend turtles a lot, because they are really, really interesting, and there are many topics and interesting topics. Of course, the origin is one of them, but each group has its own, I would say, big questions to be solved. So I recommend turtles all, like for all.
Cool. Good advice. All right. Well, thanks for coming on.
Thank you.
All right. For all the listeners out there, you can find more CheloniaCast at theturtleroom.org/CheloniaCast. We'll see you on the next discussion.